Hi, I'm Rachel Gorrell and I'm Scientific Lead of the CVL-865 programme at CVL Therapeutics, and I'm going to tell you about our journey to realise. So I've just paused on some administrative items here with a forward-looking statement, and then here are my disclosures. I wanted to start with a little of information about CVL, who were formed a couple of years ago as a partnership between Bain Capital and Pfizer. And the focus is on identification and development of selective drugs for targeted mechanisms in neurology and neuropsychiatric disorders. And a really important program for us is in the treatment of epilepsy with our drug, which has a novel selective mechanism of action. Before I dive into more detail on CVL-865, I wanted to give you a little reminder on GABA A receptor structure and benzodiazepine pharmacology. So GABA A receptors are ligand-gated ion channels and control chloride flux. The subunits assemble as a pentameric complex, most frequently consisting of 2 alpha subunits, 2 beta subunits, and a gamma subunit. And it's only when these alpha subunits are alpha 1, 2, 3, or 5 can there be formation of a benzodiazepine binding site, which sits here between the interface of the alpha and gamma subunit. Benzodiazepines do not have intrinsic efficacy, however, they are positive allosteric modulators and enhance the effect of GABA non-selectively at those receptors containing either alpha 1, 2, 3, or 5 subunits. And this GABA-induced chloride flux hyperpolarizes neurons and prevents action potentials and dampens down neuronal excitability. Now, this dampening down of neuronal excitability makes benzodiazepines efficacious drugs in a range of indications. However, their use and their dose is limited by adverse events, even at low receptor occupancy. Now, understanding of benzodiazepine pharmacology and GABA receptors has become more comprehensive over the last 20 or so years. And this has been in part due to the development of alpha subunit point mutated mice, in which there's a histidine to arginine molecular switch which ablates drug binding. So in effect, you can develop alpha 1 knockouts, alpha 2 combinations, and so on. These mice, together with subtype selected PAMs, has led to a mechanistic understanding of the pharmacology of benzodiazepines, and this is shown in the table at the bottom. So, for example, the anticonvulsant effects of benzodiazepines have been attributed in part to alpha 1 subunits. However, those alpha 1 subunit containing receptors have also been shown to be responsible for many of the undesired effects of benzodiazepines, including sedation, cognitive impairment, and addiction. The alpha 2 subunits have been shown to be responsible in part to the anticonvulsant activity of benzodiazepines, but also some of the other desired properties of benzos, such as analgesia and anxiolysis. So the hypothesis at the start of this program was that if you could develop an alpha 1 sparing positive allosteric modulator, it would enable clinical exploration at higher receptor occupancy than a classical benzodiazepine, have an improved adverse event profile, but also retain that broad spectrum and robust anticonvulsant activity. And this is where the CVL-865 program came from. Now I'm going to jump straight into some of the efficacy data we have with CVL-865. And on the left here is an example of some of the preclinical efficacy data. And this data shows broad spectrum anticonvulsant activity in a range of models. This includes in the genetic absence epilepsy model, in the PTZ, and also the amygdala kindling, which is shown in the graph in the bottom. So here you can see that increasing doses of CVL-865 protect those animals from seizures. And in general, in those models, you would need to achieve around 50% or more receptor occupancy in order to see efficacy. On the right-hand side is data from a photosensitive epilepsy clinical trial, which was run a couple of years ago, and I presented previously at that meeting in detail about that study. So in this study, we have subjects with photosensitive epilepsy, and this was designed as a crossover study. So subjects received either placebo, a low dose of CVL-865, which achieved around 60% receptor occupancy, a high dose, which achieved around 80% receptor occupancy, and lorazepam as a 2 mg dose as our positive control. And in purple here, you can see the placebo dosing. Here you can see that there's robust reproducible photosensitivity observed when those subjects were dosed placebo. However, both doses of CVL-865 and also the positive control resulted in a robust reduction in photosensitivity. And in fact, we saw full suppression in 6 out of 7 subjects. So this accumulation of both preclinical and clinical data to date has demonstrated the anticonvulsant activity of CVL-865 is retained, and this is potentially through that high receptor occupancy being achieved at alpha-2 subunits. What about the safety profile? So we do have a lot of safety data with CVL-865, and I'm showing data here from the last multiple ascending dose study that we did. And in that study, we used a quick titration during that 1 week. This was followed by 2 weeks of maintenance dosing and then the follow-up period. And in that trial, we had 2 cohorts, the first of which achieved 25 mg BID, which is around 80% receptor occupancy, and the second cohort, 42.5 mg BID, which achieved more than 80% receptor occupancy. And the table shows the most frequent adverse events that were observed in that trial. And these adverse events were mild and they were limited. And so you can see here, for example, the dizziness was reported in 1 subject during the maintenance phase and 2 subjects for the 25 mg dose group and 1 subject during the maintenance phase, the first week of the maintenance phase. And dizziness was reported in 3 subjects in that titration phase and then 1 subject in maintenance, but also in follow-up. For somnolence, we had no subjects reporting somnolence by placebo, and in the low dose group, we had 3 subjects reporting mild somnolence during the titration. But in fact, when they achieved that 25 mg BID dose, there were no reports of somnolence at all. And the 42.5 mg dose group, there were no reports of somnolence either in the titration or in the maintenance phase. So this study shows the ability to achieve 80% or more receptor occupancy without seeing significant somnolence. And another interesting observation, an important observation from this trial, was that there are no effects on withdrawal. So when we abruptly discontinued CVL-865 after 3 weeks of dosing here, we saw no withdrawal symptoms reported by the subjects. And this isn't the case for benzodiazepines, where frequently you would see reports of withdrawal following abrupt discontinuation. So this again is an important differentiating factor from benzodiazepines. In the interest of time, I haven't covered all the data available with CVL-865, but there's more available in the public domain. So we have target pharmacology data and receptor occupancy. I've briefly shown you the anticonvulsant activity in the photosensitivity model, and that we're safe and well tolerated at our target dose. So really the next piece in the puzzle is a phase 2 study in drug-resistant focal onset seizures, and this is our REALISE trial. And our focus is on the potential for patients to accomplish or realise their goals. I'll briefly talk to you about that trial design. So as I mentioned, it's in adults with drug-resistant focal onset epilepsy, and they're on a stable background of anti-epileptic drugs. Our study involves a 2-week titration period, 8-week maintenance phase, and then a 3-week taper. And we have 3 arms in the trial. So we have our placebo group, we have our 7.5 mg BID group, and this dose group achieves around 60% receptor occupancy. So this is similar to the low dose in the photoepilepsy trial. And then our higher dose at 25 mg BID achieves around 80% receptor occupancy and is around that higher dose in the photoepilepsy trial as well. And those subjects that complete our 8-week maintenance phase have the opportunity to join a 57-week open label safety study. And more details are available on this study on ct.gov. To say thank you to the organisers for the invitation to speak, and I welcome any questions.